We finish up our journey through the nephron by looking at the actions at the distal convoluted tubule and the collecting duct. Now, some of the topics that we'll talk about here, we've already broached on. We've talked about hormonal activity at the kidney and how we reabsorb water and salts to help regulate blood volume and blood pressure. We've talked about the actions of the kidney at least a little bit when we talked about pH regulation and our ability to secrete hydrogen ions and bicarbonate ions at the kidney in hopes of regulating the acidic level of the blood. So now we get to look at those mechanisms just a little bit closer and close the book on those topics. Remember, we're looking at filtering out of the blood any waste products and anything that we have in excess. That filtration is going to take place at the glomerulus, our renal capsule. We produce a filtrate that goes into our proximal convoluted tubule, upon which we reabsorb approximately 65 to 70% of all of the solutes that we just got done filtering, and we reabsorb them back into the blood using mechanisms of primary and secondary active transport and facilitated diffusion. Next up, we looked at our loop of Henle and our ability to reabsorb water by osmosis and salts by active transport, and combining both of those types of transport to get a multiplying effect in our counter current multiplication concept. Now, anything that's left in our tubule at this point proceeds on to the distal convoluted tubule and collecting duct. Now, these are very distinctly different anatomical regions of the nephron, but for most of our discussion here today, we're going to be taking both of those parts and including them together. Now, the distal convoluted tubule, remember, was the part of the nephron that became important for our tubuloglomerular feedback of glomerular filtration rate. It was sodium content in the distal convoluted tubule at that point that would be detected by our receptor cells at the macula densa and change renin secretion as produced by our juxtaglomerular apparatus, therefore influencing blood pressure through the secretions of our renin-angiotensin system. Now, this will ultimately come into play for the hormonal secretions that affect the kidney as well. Remember, we had three overall processes that contributed to urine formation, and the flip side of that is going to be blood composition. Filtration at the glomerulus, reabsorption along various parts of our tubule, reabsorbing different types of solutes, sometimes water, by different mechanisms. And then finally, we get to our tubular secretions. Now, that's going to be more of a topic that we have now for our distal convoluted tubule and collecting duct. When we look at the actions of the distal convoluted tubule and collecting duct, that's where the primary focus of any hormonal activity is going to be. We're going to be looking at the effects of the three main hormones we've talked about all along: ADH, aldosterone, and ANP. Aldosterone being the main hormone that we have to increase water reabsorption, therefore increasing blood volume and increasing blood pressure. Aldosterone directly involved in the reabsorption of salts at our distal convoluted tubule. But as that salt content increases blood osmolarity, now we secondarily get a secretion of ADH to reabsorb a proportional amount of water with those salts. And then lastly, we had ANP, which is a direct antagonist to our ADH secretion, blocking any water reabsorption at our collecting duct, causing increased volumes of more dilute urine to be produced. Now, we get questions at this point from students who ask about diuretics as a medication. And it's important to realize that a diuretic is going to be a class of medication with any number of specific means by which to control that. A diuretic simply will block reabsorption of water, therefore increasing urinary volume and frequency. You're going to get that fluid off of the cardiovascular system and put it into the urine. Now, as we've already looked at some mechanisms throughout our nephron, we could see any number of sites of diuretic action. We could block water reabsorption at the proximal convoluted tubule. We could have osmotic diuretics affecting our loop of Henle. We could have varying medications that either will block ADH receptors, block ADH secretion, play a synthetic role of ANP. Any way to get water from being reabsorbed at the kidney and therefore by default spilling over into the urine. It's also worth noting at this point that we have other hormones that affect the kidney. Now, some of which are not going to be a topic of discussion currently because we have an entire other hormonal topic coming up. We'll talk about any number of hormones. But anytime we talk about an action where we're looking to increase or decrease blood composition of something based on hormonal activity, there's probably going to be a mechanism at the nephron to handle just that. And on this particular diagram, you see parathyroid hormone involved in the reabsorption of calcium to elevate blood calcium. Now, again, we haven't really talked about the mechanisms of hormones distinctly yet, but on the surface, there are going to be a messenger chemical secreted by an endocrine gland into the cardiovascular system and using the blood as a means of transport. Any cell with a receptor specific for that hormone is going to be able to use that hormone as a means of communication. We talked about homeostasis on several different occasions, obviously. Receptor, control center, effector. When we talk about the control center and its ability to recruit effector organs, we can do that by nervous system innervation, directly sending a neuron out to that target cell and innervating it to get a response. Or we can use hormonal secretion in some means, where we secrete a hormone into the circulatory system. It reaches our target cell, binds to a receptor on that cell, and intracellularly, that cell will respond to that particular hormone and bring about whatever effect we would expect that hormone to bring about. For ADH, we have a vasopressin receptor. Now, vasopressin is going to be an older term for ADH. So whenever you see vasopressin in any of the resources that you happen to look up, interchangeable with ADH. But we make use of bulk movement when it comes to ADH mechanisms. Vasopressin, or ADH, will bind to our receptor on the cell surface. Now, just to orient us, these are cells that are making up the nephron. Here's blood on one side. Here's our tubular filtrate on the other. So this is going to be the fluid that just left our loop of Henle. Cyclic AMP intracellularly triggers small vesicles inside the cell to merge with the cell membrane adjacent to the filtrate. Now, we've talked about vesicles in several contexts already. We talked about vesicles as far as granulocytes for our white blood cells, storing different chemicals as part of an immune response. We've talked about lysozymes in generic cell biology as a storage mechanism. Here we have vesicles, which are small pieces of cell membrane that distinctly have a transporter for water. It's a water channel. So when these vesicles embed themselves in the membrane, they embed a water channel. And if you look at what we have going on by way of our solute concentrations, By the time we make our way back up to the distal convoluted tubule, we are back up to a 300 milliosmol per liter filtrate, which is a lower environment than what we see intracellularly. So the intracellular fluid relative to the filtrate at that point is hypertonic. So we see water move through our water channels for means of efficiency, but we see that water move across that membrane towards that high solute concentration. Likewise, we see an even higher concentration again in the blood. So we continue that movement of solute from lower solute concentrations to higher solute concentrations as we go from the filtrate to the intracellular fluid and then later on out into the blood. So as long as we have vasopressin binding to our receptor and increased water channels, we will continue that increased rate of reabsorption of water from that collecting duct. It's important to be able to understand what happens to the blood and what happens to the urine in the presence of ADH. Essentially, what happens to one of those volumes of fluid, we'll see the opposite occur to the other. So in the presence of ADH, we reabsorb more water, increasing blood volume, but at the same time, decreasing urine volume. By reabsorbing more water into the blood, we're decreasing blood concentration, but the flip side is we're increasing urine concentration. As we reabsorb more water, now we can have implications for our blood pressure. As we put more fluid into the circulatory system, we can help elevate blood pressure. Remember, we had discussions about how osmolarity of the blood could lead to a high blood pressure due to water retention. And we would give that patient a diuretic to get that excess fluid off. Well, if we have someone with a low blood volume and a consequential low blood pressure, here's a natural mechanism that we have to bring that blood pressure up as part of a negative feedback loop. Aldosterone, as we mentioned, directly acts on sodium resorption. Now, we still have a very similar mechanism where we have a mineralocorticoid receptor as part of the cells of the distal convoluted tubule. We trigger the increased production of sodium pump outside the cell into the blood. And as a result, we see more sodium taken out of our filtrate as we create a very low sodium concentration inside our cell, dragging that sodium in and then pumping it out the back door, so to speak. Now, in this way, we will also get a glimpse of some of our secretions, because we see the exact same thing happen with potassium, just in the opposite direction. We're pumping sodium out, we're pumping potassium in, creating a high concentration inside the cell, and in fact, higher than what we see in the tubular filtrate, and therefore potassium will be secreted into that tubular filtrate. This is a diagram that had popped up in one of our previous discussions, but I want to bring it up there again because at the time we said, ADH and aldosterone, we would get into that a little bit closer once we looked at the structure of the nephron. But now it's important that we have to start tying mechanisms together. And it's important that at this point, for this week, you have to tie in blood volume with blood pressure, because you will get questions on the next exam regarding that topic. Even though blood pressure is not really something we're getting into directly this week, it's something we've already discussed, and now we're just implementing ADH and aldosterone as a way of correcting that mechanism. So if we see a hypotension, for example, or a decrease in sodium getting filtered at our glomerulus, our tubuloglomerular feedback is going to secrete renin. which ultimately increases production of angiotensin 1, increases production of angiotensin 2, cause our adrenal cortex and our pituitary to secrete ADH and aldosterone. We retain fluid, we retain sodium, that's going to increase blood volume, and we always want to think about that relationship of pressure and volume when it comes to a fluid. If we increase the volume, we also have the opportunity to increase the pressure. So you will have to be aware of the role of ADH when it comes to regulating not just blood volume, but to carry that one step further into how it regulates blood pressure. Now, our tubular secretion we'd already mentioned when it comes to, our sodium and potassium exchange, secreting potassium into that tubular fluid and ultimately having it carry on out into our urine. But of course, we also see a role here when it comes to pH regulation. We can secrete hydrogen ions by way of another pump, our sodium hydrogen pump. If pH gets too low, if the blood becomes too acidic, we can activate these pumps, dragging hydrogen out of the blood and dumping it in our tubular fluid to be let go into the urine. Likewise, we can increase reabsorption of our bicarbonate ions. Now, we see a direct connection now back into our carbonic acid buffer. We had brought that up previously when it came to respiration. We distinctly looked at carbon dioxide as far as it being in an equilibrium with our carbonic acid and therefore our sodium bicarbonate buffer. Now we see the kidneys jump into that exact same equilibrium. Our respiratory system manipulated it by way of manipulating carbon dioxide levels. Our nephron is going to jump into it by regulating directly the amount of hydrogen ions present. So now we see a direct connection and sharing of a role, if you will, between our respiratory system and our kidney when it comes to regulating blood pH. If we see an increase in hydrogen ions added to the blood, we will then see a mechanism where our sodium hydrogen pump pumps hydrogen out of the blood and putting it into the urine, while at the same time we see an increased respiration rate, expelling off additional CO2, decreasing the amount of carbonic acid, therefore decreasing the amount of hydrogen ions. So both of those systems play a role, a shared role, in regulating blood pH. We can help maintain our bicarbonate reserve in the blood by reabsorbing our carbonate, giving us ample chemical mechanism to run our buffer. Likewise, if we see the blood become too basic, too few hydrogen ions, we shut down our sodium hydrogen pump now, helping us reabsorb as much hydrogen as possible, or at least retain what we have, secreting out our bicarbonate ions, this time decreasing our respiratory rate, preserving CO2 in the blood, which preserves our carbonic acid and therefore preserves more of the hydrogen ions, trying to get that pH back down. So we're really kind of cluing things up here. And this week's lecture is not a particularly long one, as you we'll tell here in just a moment. But it is important for you to go back and look at your previous sections now. Because while this lecture might not be heavy in new material, it does create the awareness of the interrelations with those other systems. So please go back and integrate what we learned about blood pressure and why regulation of blood volume is going to be important in regulating that blood pressure. Go back and look at how the respiration rate plays into your bicarbonate buffer. and now add the kidneys mechanism onto that pH regulation. Now, most people are going to struggle a little bit bringing all that together. So it's important that if you look at that system and you have trouble interrelating both of those topics, please reach out and we can go through it. But consider that part of the exercise is noodling your way through that process. So as far as key concepts go, we kind of look at key concepts for the entire urinary system right now. Obviously, anatomical aside. But by the time we got to the nephron, it's important to understand the role of the hormonal action. Aldosterone, ADH, ANP. And then looking at the role of the nephron as a whole and the actions that we see at the proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct as a whole, as far as what the nephron does to accomplish both blood composition and urine production through the removal of waste products from the blood, the regulation of blood volume, regulation of blood solutes, and the regulation of blood pH.